Welcome everyone to this webinar on Shall We Model the Brain? I'm Priel with MetaCell. I'm the EVP of Client Relations. I'm joined by our panelists who I will let them introduce themselves. If you want to start, Anton.
Sure. So hello everyone. My name is Anton LeBouff. I'm an associate investigator at the Allen Institute for Brain Science in Seattle. So my team here does work on data analysis and modeling, biologically realistic modeling of brain circuits.
Thank you, Anton. Sean?
Yeah, I'm Sean Hill. I'm the director of the Krembil Center for Neuroinformatics here in Toronto, Canada at CAMH. It's the Center for Addiction and Mental Health. And the team here really uses multiscale modeling to attack the challenge of understanding mental health disorders.
Great, thank you. Kimberly?
Hi, I'm Kimberly. I'm a postdoctoral fellow at Harvard Medical School, and perhaps will represent the experimentalist side, but also we use models to help understand what's happening at the level of neural circuits.
Great, thank you. And lastly, Bill.
Bill Litton. I'm at State University of New York in Brooklyn, SUNY Downstate. I'm a professor there of neurology and physiology. So I see patients, and primary activity is modeling neurological and psychiatric diseases.
Great, thank you. And audience members, please, we encourage you to share questions. We want to make this as interactive as possible. So go ahead and ask questions, and I will field them to the panelists as they come in. So we'll first again thank you everyone for joining us today. Thank you for the introductions. I think the first question really to anyone is, what do you all think of brain simulation science?
I think one of the problems with brain simulation science is that there have been so many different versions of it, and when you compare what we're doing in the brain to what other organ system modeling is doing, we tend to be a bit both ahead and behind, and we have certainly some unique issues with regard to the brain being an organ that has information in it. But because of all that focus on information and calculation, and the brain as a machine, and the idea that, yes, you could reduce it all to silicon and upload your brain and all kinds of other things, it's kind of distracted from the, to my mind, real science, quote unquote, of looking at multiscale modeling of the brain as an organ system, which also happens to think and calculate and remember and such.
Okay, so I know you, is there something, like, what is the difference in the types of modeling that I guess you do, or what is missing from that type?
Well, so multi-scale modeling, which I think I do, Sean does, Anton does, really trying to reach across the scales, understand how cells work, how ion channels work, in some cases understand how molecular mechanisms work, whether in the synapses or cell body, or affecting the ion channels through neuromodulators, and then up to cell level, or really dendritic level, then cell level, then network level, then area level. Those would be the multiple scales. The efforts on the, for example, the engineering side, which are phenomenally successful, so you can't begrudge them that, now with deep learning, kind of short circuit all that and say, okay, we have a hypothesis as to how neurons work together, which really is similar to the hypothesis that came out of Hubel and Wiesel many decades ago, and we're going to make that work for us, and it does work for us, but it's really missing a lot of what's important, certainly for neurological disease. In fact, I don't think there's anything in what they do that can help us with understanding neurological disease, and I think also for just brain function, it's just a very limited perspective. Another perspective that I could mention is just trying to do things in more closed forms. For example, again, but better not to mention names, that's kind of mean, but some people have the idea that we're going to get the grand unified theory of the brain, and I'm skeptical of that. Different brains, different animals, different levels of development, different levels of brain degeneration are very different. Thalamus is very different from cortex, different from basal ganglia. So there's just so much biology there that you just kind of elide and miss if you try to pull it all together.
I think that speaking as an experimentalist, modeling is becoming inevitable as you try to understand circuits. There has been a debate in the field for some time about whether models are useful or whether one should try to put all the information into your head and have some insight. And that's certainly important, but I think it's becoming more and more clear to us that when you're looking at the communication of many populations of neurons, it's just too much to put into a single person's head. So models are truly inevitable in neuroscience, I think, because we are dealing with a complex system.
I think there's another side to it, which is really important, which is that just through the act of starting to build models, you change the way in which you measure, and you think differently about the types of data that are essential to measure, and how would you measure the right kinds of data. And I think that partnership between modeling and experiment is so critical to making the scientific process more efficient, right? And just understanding what's essential to measure and which things can be predicted quite readily from the models.
Great, thank you, Sean.
Yeah, very much agree with everything that was just mentioned. I'd like to add a quote which, for me is really inspirational, and I think it really describes what many of us are trying to do in brain modeling, and that is a quote from Richard Feynman, who famously said, "What I cannot create, I do not understand." Right, and so the idea here is that we collect a lot of different data, but how it all comes together, we have some ideas about how things may work, but are these ideas correct, and when they are correct, and when things break down. So because of all of that, like Kimberly said, it's really important to put things together into models and try them out. Another thing I want to mention though is that while I'm really passionate about brain simulations, I think also, like with many other things, they have a great promise, right? I think if we make brain simulations work really well, it will provide tremendous scientific progress and potentially also practical progress. But time will tell. Time will tell. I'm optimistic, but we are not there yet.
Thank you, Anton.
One of the challenges is that I think very often when people hear that you're doing brain simulation or brain models, that there's an assumption that therefore you know everything about the system, you understand it all, and that's why you're building a model of it. And I think it's quite the opposite. I think it's a way of understanding the data and understanding the relationships between the different physical scales of the biophysical entity of the brain. And it's a tool. It's a tool for understanding. So I think it's really important to see that as one of the primary motivations. It's not because we understand it all that we model it. It's because we want to understand it.
Absolutely.
To amplify that, I'd say it's great that to have experimentalists work extremely closely with modelers, and often it's the case that we find out as we're doing the modeler that there's some key parameter that hasn't been measured or hasn't been adequately measured, and go back and check that and put it in the model. And it's a process of gradual improvement, like any large model, like a model of an internal combustion engine, models of the weather. Gradually improving how you do it. You don't know anything about either.
Absolutely, and I think, part of our challenge in communicating has been that there seem to be two goals in modeling. One is to understand the brain as it exists, and the other is for the application side. And obviously AI has jumped forward in terms of applications with deep learning and can do really amazing things. So how we communicate about the fact that neural-based models are still critical if we want to cure disease, as you mentioned, but that is a separate and equally important goal. But it's not the goal of just jumping to the engineering side.
Mm-hmm. So we actually have a question from Frances Jeanson. She asks, how should we validate a simulation model to be used for informing new medical treatments?
It's a really great question, and again, it depends on what level you're actually thinking of a treatment, because there are many potential levels. From behavioral level, cognitive behavioral therapy, down to specific compounds that are acting on subcellular signaling pathways. So I think one level you have to have a clear sense of, well, where are you trying to target the treatment? But I think that, again, the key thing is you can't have a model that only replicates one set of experimental findings if it's going to inform as a treatment. I don't know if maybe we can mute the other microphones, because there's some kind of loud noise. But I would just emphasize that building models that can replicate multiple different experimental findings at different scales increases your confidence in their relevance to answering specific questions like treatment.
Bill, can you try muting yourself?
I was muted.
Oh, okay. Oh, there we go. Thank you, Sean. So we actually got another question from Jay Coggin.
I'd like to amplify that thought that Sean had. So I think we have a lot of distance to go compared to other organ systems. Really, in orthopedics, for example, these systems are already being used. There's already discussions with the FDA about how best to validate. And it depends also on the severity of the disease. So I have a colleague, Andrew Knox, who's working as a child neurologist directly with very severely impaired children with epilepsy. And he's, I think in the next 5 to 10 years, will be ready to use the modeling directly for individual personalized medicine on these kids, that because they're so severely impaired, you can have to try something. But yeah, validation is extremely obviously important. Verification, validation, and difficult.
Great, thank you, Bill. Does anyone else have input?
Well, I'll just add that, in addition to all these aspects that have been mentioned, I think basic fundamental research is important, right? So it's essential to have a good baseline. If we want to understand how things go wrong, we also should understand how the system works when things are sort of normal. Applications are very important. For the applications to make good progress, it's essential to build this very basic foundation and understand the system in its normal state. And all the intricacies of that system, like Sean said, it's essential not to just build a model that does only one thing, because the brain is very complex and it can do many, many different things. So ideally, the model would be multi-purpose.
Thank you, Anton. So actually building on that, we have a question from Jay Coggin. While we've really just begun, how will we know when we are done modeling the brain? I think this points to not being a singular point of focus, but like you said,
never.
Never. Okay.
Yeah, I would concur. I don't think that I don't think you're ever really done. I don't think we're ever going to have a complete model of every aspect of the brain. And I think part of the whole point of modeling is to continue to explore many different aspects of functioning and over different time scales and across different spatial scales. And that, the goal in my mind is not that we'll have a complete model of every aspect of the brain always, but that we'll continue to use modeling to enrich our understanding of many, many different phenomena.
Great, thank you, Sean.
Science is the endless frontier.
All right. I guess the next question I have is, what would you all point to in the last 10 years in brain simulation science that has been valuable? What has excited you?
I could point to two things. I think one, and of course I'm totally biased, but one thing was that in the Blue Brain project, I think one of the things that we did learn was that you could take a set of biological measurements of a piece of cortical tissue. You could assemble that into an integral data-driven computer model, and that would give rise to, without tuning for specific network behavior, it would give rise to behavior that was consistent with a biological entity. So from my perspective, that was a very important proof of principle, that integrating lots of biophysical measurements from the lab would actually be informative about emergent level behavior in the circuit. And I think that was an essential discovery. The other element of it is that we also learned how do you actually organize data in a way that it can inform those models. And that's another very fundamental, very critical discovery.
I would say that most of the progress in the past 20, 30 years has been on the technical side, and certainly the Blue Brain Project as well as the Allen Institute have really led the way. Another person I mentioned would be Roger Traub. One thing I would put before Sean, the models you're referring to are fantastic and have been and are going to continue to be extremely influential. But I wonder what emergent properties were seen in the Blue Brain model that was, for example, different or better or more precise or more accurate than what would be seen in the large Traub cortical model, or some of these, maybe Pacha and Diesman, though I think that one might be too simple. It's hard still to find where the individual advances are in particular models, and maybe Sean will correct me here, but I feel that we still don't have enough good measurements either in our models in terms of very high order correlations, or in, more importantly, the real brain data in terms of that kind of correlation, that kind of thing, which we can really match up very precisely.
Yeah, I think it's an important question, and I would also emphasize that it's by no means like complete and finished, and there's a long road to go. But I think that what fundamentally we were able to do was really anchoring the constraints of the parameters off of laboratory data. And then
no, I agree. It was a better model. I agree.
And so, well, from that perspective, I mean, because I think previously, and certainly, I'm somebody who has built models before that were all very much hand-tuned to create a network phenomenon, right? And that's very commonly the approach, right? Is that you sort of these ingredients should all fit together, and they should give rise to this oscillation, or they should give rise to sleep and waking, stuff that I've done before. But the interesting thing for me was we could really feed biophysical level measurements into data-driven pipelines, and it would give rise to phenomena that we did know existed, low frequency oscillations, and then by changing a single biophysical parameter, extracellular calcium concentration, for example, we could see that the circuit changed state. It changed its integrative properties significantly in a way that was consistent with the biology. And so going from a synchronous to an asynchronous state through, again, measurable biophysical parameters. We always knew theoretically this space, but we didn't have an integration of data including cell type specific models and this type of thing that would give rise to that. And I think conceptually, because there had often been an argument about, well, sure, you can find anything in simplified networks, or you can find anything in complex networks. Constraining it to the data, constraining it to the biophysics, constraining it to the measurable level was valuable in sort of proving that proof of concept that you could do it and that it would give rise to something useful.
Great. Kimberly, I'd love to get your input as an experimentalist.
Yeah, well, this actually raised another thought in my mind, which is, something probably both Alan and Blue Brain can speak to, that once you have a model that shows something interesting, how you communicate that effectively to other scientists is, I think, a huge challenge, because we're used to papers where a single finding can be put into a figure, right? And you see some change. Oh, okay, this gets higher, this gets lower, and you can look at the figure and see that. But a model is much more complex, and really to convince people that what you've done is valuable, you'd like them to be able to play with the model, right? To see that for themselves, because these models are high dimensional, and I mean, and I think that is one of the areas where we have most struggled as a field, not just for these large models, but even for simple models, the kinds that I do as an experimentalist, where it's, fitting some coefficients, right? But even these are complex enough that communication is a real challenge. And so I think we need to somehow move beyond the published paper, physical paper format, to a way that we can share models so that people can really appreciate the complexities that they're trying to communicate.
Yeah, this is an excellent point. So, I think there are several things that people are trying to do to address it. But first of all, I think it's very, very important to share everything, be as transparent as possible and serve everything to the community. So that's, for example, why the Blue Brain and us, the Allen Institute, we partnered together. And then also other people joined. So, for example, Bill and Salvador in his team joined as well, and other people joined in developing the file format. We call it Sonata file format for sharing models and simulations. So, just simple things like that. There are existing formats to do that, but not that they don't necessarily cover all the different bases, or they might not necessarily be very efficient for the large simulations like the types that we do. So anyway, we had to develop that. We share all our models. So for example, the Allen Institute is sort of well known for sharing, gene expression atlases and other types of experimental data. But now we also share the models that we developed. In fact, we make them available to the community even before publication. So I think these are all good steps. There are challenges there, of course, in that you can share the model, but then how do people simulate it? And then, it's not just about running a simulation, it's about understanding it. So it has to be some kind of iterative process where experimentalists gradually become comfortable also with, maybe trying to run some simulations. An additional challenge there is that some of those simulations are extremely computationally expensive, right? So you really need a huge allocation on a supercomputer or something to run such a simulation. The neural network community, the artificial intelligence community, made huge strides because a lot of the computations can be done on a single GPU under your desk. And there are some opportunities here for large-scale brain simulations as well. Maybe you don't have to do everything with biophysical level of detail. You can do point neuron simulations, for example. They can fit on a single CPU for a pretty sizable system. The last thing I wanted to mention here is that papers are still extremely important. In the end, it's important to share models. It's important to be able to play around with them and do different things. But in the end, it's very, very essential to do good science and make specific discoveries. And so in our case, for example, we sort of were doing a thing that's similar to what Blue Brain has done, but for the visual cortex of the mouse. And when we put together the model, we went after very specific experimentally observed properties, such as direction selectivity of neurons, that were recorded in vivo by our colleagues, experimentalists. And we were asking, well, okay, we have this model, given all the biological constraints, how can we reproduce these experimental observations? And in the process of doing so, we came up with some predictions for the structure of the circuits that can now be tested experimentally, and some of them have been confirmed already. So I believe this is really how it has to go in an iterative process, going back and forth between the model and experiment.
I wonder if, oh, sorry, go ahead.
Yeah, Mark Harold, about 10 plus, maybe 20 years ago, he's at Argonne National Labs. He wrote a nice article about how we really eventually will need, and I think we do now need, to couple, to create interactive supercomputing. And I think we're kind of on the verge of that. Some of the software we're working on, as well as Alan and Blue Brain, is going to move towards that. It's not all things that are going to run on your mobile phone. Some of these things do require HPCs, but you really do need to interact with them, and it does have to get back to the experimentalists, and I think we can make that happen.
I wonder if one effective approach in the meantime would also be to somehow set up fellowships, perhaps, for experimentalists to come and use a model, or the way that telescope time is allocated right now in astronomy, right? You apply, you have a grant, you say, I'd like to use this telescope for a month, right? You go, you aren't obviously technically proficient in the telescope yourself, you're the experimentalist, but you come and you know what experiment you want to do, and you work with the people who know the technology to run an experiment, right? And I wonder if for these really large scale models, we might be able to implement something like that, where experimentalists could apply to run experiments and could go physically and help make that possible. I mean, that would be very exciting for me as an experimentalist.
So that's actually something that Blue Brain is doing with some experimentalists, and Human Brain Project has sort of made a part of their effort around having these resources available that you can apply to run things. I think it's a very good model. I think it's a very good point, and I think that kind of that virtual observatory for simulations is a great model, but it does require still speaking the same language and understanding between the two worlds. And I think one of the interesting things is that you need this common tools at the analysis level to make comparable analyses. So for example, if you're looking at calcium imaging, wouldn't it be great to do in silico calcium imaging so that you're really comparing the same kind of modalities? Similarly, spike train, spike analysis, intracellular. So I think very often the experimenter has a set of tools that they're used to using, and that gives their perspective on this very complex system. And enabling a similar comparison is actually quite key to being able to work together.
Kimberly, you're invited to come visit anytime you want. Just let us know.
Oh, wonderful. I will take you up on that. Thank you.
Very, very nice. So we actually got another question from an audience member. Dario Dollar asks, just kind of switching gears a little bit, what fraction of the brain function do you expect will be translatable from preclinical species to human? How much of it translates across preclinical species, for example, mice to rats, or even between different gender animals of the same species?
It probably depends on the disease. I mean, schizophrenia, of course, has been much harder to handle than, for example, epilepsy, because a lot of the manifestations are in the area of language and cognition, which may or may not take place in some form frusta in these animals, but certainly is not something we can access. So, different brain areas, different diseases, different problems, different solutions.
Good point, yeah.
Yeah, I think it really, yeah, please go ahead.
No, Sean, go ahead.
No, I was just going to say, I mean, it depends very much on what it is you want to be able to say and ask, and it has, it's a parallel discussion as to whether or not animal models of disease are relevant to understanding human disease. You really have to look at very specific questions at the appropriate scale and mechanism to understand whether or not it's actually translatable or relatable across species.
That's something we just actually wrote up in our discussion of a paper on schizophrenia, that, yeah, our model is incomplete, our computer model is lacking things, but of course all the animal models are incomplete, all the animal models are lacking things, so we need to work together.
Yep.
Yeah, and another thing is that, the question was about brain function, right? So brain function is extremely diverse, and there are so many different aspects to it that may or may not include disease. But I think it's important also to take things in a perspective, to think that, well, what is the most salient thing about our own brains? Well, it actually makes us feel, it makes us be conscious, right? So that is a huge part of the brain function that we are not even typically considering when we build brain models. What about consciousness? What about perception? What about how it is to have a brain, right? So that, I think that throws us back to the question about when we are going to be done. There is a lot to do here. There's a lot to do. There's some of the things we don't even have a little sliver of understanding.
Come back in a few millennia, then we'll answer the question.
But that does raise a point that I wanted to raise, which I just deeply believe the importance of, which is modeling different states in a given circuit. And I think that part of the reality of any given brain circuit is that it's in a different condition based on its neuromodulatory condition, and the way that it integrates information changes dramatically, and the way it responds to stimuli, and the way that the whole system functions, the interaction between excitatory and inhibitory cells, the synaptic dynamics, everything gets modulated. And I think that one of the most valuable things that I personally have seen is the value of modeling different states within the same circuit in terms of understanding and constraining the model, because it's totally non-trivial that one given parameterization is going to capture the transition between different states.
Great.
And I think I think we're really at a cusp where more and more experimentalists, the kids younger than me, they have the coding ability, right, to step up and bring both the modeling and the experiments. And it's really that, I think Sean is right, it's a shared language issue, which I guess is a problem all intersectional fields face in the beginning. And I think we're really starting to get there in systems neuroscience and across the levels in neuroscience, where people have this shared language to make it happen. So I'm really optimistic ultimately. Yeah.
Great. So we actually have another question from an audience member. Dario Dollar asks, what do you think is the role of chemical biologists or medicinal chemists aiming to understand how the brain works? So as we were talking about how it's intersectional.
I guess it'll be hard to get the feedback. Is it a reference to maybe molecular dynamics type of modeling? Certainly chemistry would be very important at that very low level of modeling. At the higher level, generally we think in terms of pharmacology.
I'd say, yeah, both.
Yeah, I mean, so the notion of multiscale modeling implies that one needs to get from the molecular, because the pharmacological agents are our main ways of combating disease, up to clinical phenomenology, which may be EEG, which may be symptomatology of what's bothering the patient. And so the molecular level is absolutely critical.
I think it's critical, but I think it also poses an amazing challenge given the non-specificity of most pharmacologic agents, and very often I'm asked, well, can't you just model the effect of this drug on a neural circuit? And of course the challenge is as well, if we knew where this chemical was actually, where all of the sites of action are, what are the mechanisms of action, how is it actually impacting the system. But the reality, and especially, even in the experimental context, this, that it's very non-specific very often, that a given agent, even if you think that it's acting specifically only on a particular class of ion channel, for example, rarely is it actually as specific as you would like. And so that's one of the big challenges, is understanding really where are these compounds acting.
I think that's one of the advantages of computer modeling, is that one can make a list for many of these drugs, as well as for neuromodulators, which are also very, quote unquote, dirty in terms of their specificity of action. But then including many of those, you can find unexpected, emergent things that come out of, well, why would you be hitting all of these different places with a neuromodulator? Or why is this drug effective that hits 5 different places, 2 of which seem to be contradicting each other in terms of the effect? So I think it's a great opportunity.
Yeah.
Can I ask a question to the group? Something that I'm curious for your thoughts. In contemplating starting a lab one day, I wonder, what do you think is the right ratio of experimentalists to modelers to people trying to make those models accessible to the community? And how do we, as more, in a more academic space outside of these really large collaborations and large projects, how do we make that ratio a reality in terms of, how we structure our labs? I wonder if you have thoughts. Yeah.
Well, I just started a center here in Toronto, so I have one perspective on it. I don't think it's the only answer in any sense, but I do think, at least here in the hospital, we have a major team dedicated to just the informatics, just to how do you manage the data, how do you capture the data, how does that serve the clinic well, and how do you make sure that it's high quality, all of those things. And there's a dedicated team to that engineering. And then each of the scientists at this center, their mandate is, they're actually not going and collecting the data themselves, but they're collaborating one-to-one or one-to-many with other groups in the clinic or in the wet lab to get the data that they need. So I think, you need to have really dedicated resources, and I think at least 50-50 between modeling and experiment, but you need enough of that extra support for how do you really properly manage the data, how do you properly support those teams so that they don't get bogged down with all of the challenges of the organization, the management, the use of the data, and the dissemination.
Yeah, Sean has certainly a particular advantage there in putting together a center. It would be tough to get all the aspects that you might ideally want into a lab, particularly a startup lab. The question has a lot to do with the logistics of funding and logistics and the sociology of science as to what you can fit into a department, what you can sell in your grants. And so it's really a big picture question, which I admire Sean and Blue Brain Project for really taking that on.
Yeah. So very true. I think for someone who starts their own lab in academia, the reality is that you really have to be focused on a very specific question and sort of almost try to be orthogonal to everyone else in addressing this question, so that you can stand out, so that you can get grants, so that you can get publications. That's sort of a nice thing about academic science, but also an unfortunate thing to some extent. From that point of view, realistically, you probably may want to have majority of the resources dedicated to something that is very, reliable, something that can be is going to deliver, right? So probably the experimental side. But maybe it would make sense to have a small fraction of resources, let's say 1 person in the group, or maybe 2 people, to be computational people. And I think the best way for a person starting their own lab, especially going from the experiment, is to have collaborations. So, you know, collaborate with some of us, and then you have, you know, have a big player in the field who does some simulations for you, for example. That can be really, you know, very useful.
Cool. So we have a question from Joanna Frost. It's kind of switching gears again. In multi-scale models, we are including subcellular mechanisms to large network connectivity scale. Although understanding these complex models become difficult, what are the theoretical tools in mathematics which can be used to understand what features are transferred / important between levels of the model? And possibly use these, sorry.
One question that comes up is whether you can do, particularly in neuroscience modeling, but to some extent in other organs as well, an embedding of that type. The easiest would be, as the simplified, let's say, integrated fire modelers do, is if you can say the neuron really just does this, and we can now encapsulate it in a smaller 2-dimensional, 4-dimensional, do a phase plane analysis, activity system that then can be embedded in these larger models. The argument I use against that approach is particularly the apical dendrite of neocortical cells or of hippocampus, or the enormous dendrites of Purkinje cells, where to my mind it appears that the subcellular level, the dendrites, are interacting in some very complicated ways with the network level as it is manifest in multiple layers of cortex. So there are tools for dimensional reduction, and certainly I know people like Stefan Mihalis at Allen have been working on those, but I'm not entirely convinced that's the right approach to take. I'm not convinced it's not either, so maybe it is.
Great, thank you, Bill.
Yeah, I think in terms of tools for analysis, it's interesting actually. My experience, and I think experience of other modelers here at the Allen Institute, is that we tend to end up with more or less the same tools for analyzing our simulation data as we do for the experimental data. I mean, maybe it's just a matter of giving it some time, and something new will emerge in analyzing the simulation data, some particularly new ways of looking at those types of data. But up to now, it hasn't happened, at least for us. And, you know, we end up using the same metrics that people use for the experiment, which in principle, I think is a good thing, right? You know, you want to be able to measure the same thing in the simulation and the experiment. And the biggest difference with the simulation is that instead of an access to, you know, 10 neurons or 100 neurons, you have an access to 100,000 at a time. So sometimes it's just a matter of making those existing analysis methods scalable, or parallelize them so that you can actually run that analysis. But experimentalists are running into the same issue too, because they are recording from more and more neurons, right? You know, nowadays people record from thousands of neurons. There are papers recording from 1 million neurons at a time. So yeah, I think those issues are very similar actually.
I think one of the challenges is, I mean, there's always the question as to whether or not you've captured sufficiently the phenomena from a lower scale at the higher level scale. So for example, in modeling ion channel kinetics, if you're looking at the individual ion channel opening and closing dynamics and the aggregate, do you capture that sufficiently with your Hodgkin-Huxley style model, which kind of abstracts that to another scale? And the same thing happens as you move, say, from the cellular scale to a model of an EEG or a neural mass. You're leaving out details because you don't consider them to be causally important, and you always have to check that point. You always have to validate that. You always have to question that. And I think that it depends on the question you're asking as to whether or not it's relevant or not. So I think that's always at least from my perspective, the thing that you have to check is, are you able to recreate the key phenomena necessary, and if not, go back and recheck your assumptions, which are that you can simplify, and are you doing a sufficient simplification or a sufficiently accurate simplification to actually meaningfully capture the behavior. And again, that's going to vary depending on the particular question you have in mind.
An example from a recent result at Blue Brain I find very interesting, that it calls into question the use of Q10, which is the notion that you can alter the kinetics of an ion channel, in this case in particular, with temperature. And they have noted that the Q10 is probably different for ion channels in different voltage regimes, and really, that the notion of Q10 is really not entirely accurate, is not accurate for ion channels. And this is now a very basic thing, a very basic experimental thing that I think ought to be included as we go forward in our models.
Great, thank you, Bill. I guess we'll switch gears. I know we have about 10 minutes left, so audience members, still please do keep the questions coming. Here's a new question, everyone. How much modeling of the brain is done on healthy brains versus modeling disease?
Well, again, a lot of this.
Bill, please go ahead.
Oh, sorry, I just was going to say that I think one of the issues has been this notion of that we know what a healthy brain does. So a lot of the very basic model from an informatics perspective, which I called into doubt in my early comments, has these expectations of a brain being a particular monolithic thing that we can understand. So I think that all modeling has to take into account variability, and that variability, certainly one sees this very clearly in psychiatric disease, is among individuals always can verge off into what becomes pathology depending on the particular environment a person is raised in. And so when you're working with brains, it's really sometimes a bit hard to say. Some things obviously are really pathological, some things are not, but we're always working with this diversity.
Yeah, I think that's an extremely important point. Very often, I mean, if you're building a model of a small circuit, there's a billion ways in which you can create epilepsy, for example, right? It's the easiest thing to model in a general sense, but understanding whether or not you're modeling a specific form of epilepsy requires understanding, can you model a healthy system and then the specific change in that system that creates the epilepsy? And so, the same thing happens and it gets far, far more complicated, just as Bill was saying, when you think about whole brains and individuals and understanding the unique history of an individual and what led to their particular disorder or brain disease. And so I think, as always with this multi-scale challenge, you've got to understand, well, what is the question and at which level is best to address that question, and take into account, certainly, the bulk of the work, I believe, has been really around, can we build reasonable models of healthy conditions and then use those to explore various diseases or disorders.
Thank you, Sean. So I think I'll probably switch gears again to some high level, I guess, academic wide questions, as in, what is a funding strategy, or should that change, right? How grants are written in terms of, when people apply, is that an impediment to kind of us learning about, the brain and modeling things, just because of resource allocation and funds and how expensive these things are?
It's certainly a limitation. We in particular are trying to work very closely with NIBIB, which is one of the US NIH centers. In fact, we have a meeting coming up on these topics immediately after the SFN, the Society for Neuroscience meeting. But my feeling has been, and I think I expressed this early on in our conversation, that the modeling of brain should be done allied more closely with the modeling of other organ systems, and certainly that's the approach that NIBIB is taking, and not be viewed as something really of a different order, even though the organ itself is, yes, much more fancy and does cool things that the liver doesn't do.
Gotcha.
Yeah, I think there are certainly substantial challenges that are probably somewhat holding the brain model and brain simulation field back to some extent. On the other hand, generally, I think there is this opinion that, well, we really have to do modeling. So if you look at the descriptions of the Brain Initiative program in the United States, for example, modeling and computation and biologically realistic modeling, at least of the brain circuits, is written there, in large font in many places. So it's certainly there. It's certainly already encouraged by the funding agencies. But I think to some extent, the culture in the field has to change, because I still feel like, it's the modeling and a biorealistic modeling is not appreciated enough. I mean, part of it is, of course, on us. We need to show more progress and more value, but we are trying, and there's some iterations between us making progress and the field trying to embrace that progress. It will probably come to the better place. Well, another aspect of it, of course, is that modeling, especially biorealistic modeling, is very, very expensive, computationally expensive. So that has to be also taken into account. Probably the traditional R01 is not the best funding strategy, and so one needs to think about bigger collaborative efforts that involve, substantial expense for computation.
I think that's true in the rest of biomedicine as well. There's more appreciation for the need for massive collaboration projects of the kind that the physicists have illustrated at CERN and other places, where if you have a paper, it may have 1,000 authors on it, because everything is so complicated that we need complicated social structures to attack them.
Yeah,
I think the more that we can do also in the open source, and the more that we can share even the little steps that we take individually. And I really think it is a culture issue. For example, I've built a little behavior rig that runs on Arduinos, and I'm working to make sure that goes online, and all of the steps for the building the rig and doing the analysis of the behavior are spelled out so people can follow step by step. And so even these little things, right, there just needs to be more peer pressure in a way to take the work you've done and make it accessible so that people can reuse the tools that you've built. And we'll get there.
Thank you,
Kimberly. I think that's very important. I think the other side of this, which has been a challenge, is because of some of those computational challenges, being able to do models that work at the biophysical scale that the experimenters are working at, and specifically being able to speak that same language, using measurements from the lab to constrain and inform models, and to speak that same language has been a big gap, because I think some of the required simplifications, or even simplifications that people thought were critical and theoretical necessary, there's been a bit of a disconnect between the laboratory experiments and the models, and that's not always, but there, I think a lot of computational neuroscience, I think both the experimenters and the modelers would benefit from a closer interaction and closer collaboration and a deeper understanding of the biological system and the data that's coming out there, at the same time that the experimenters would benefit from modeling that would be directly integrated with their experimental work.
Great, thank you, Sean. So I know we have 2 minutes left. I'd just like to give everyone on the panel, time to, I guess, give final thoughts or comments, and then I think we'll wrap it up. So whoever would like to go.
Well, maybe I'll just express my opinion about the role that I think the brain modeling could play. Ultimately, I think the best role for it that I see is a platform for thinking and hypothesis testing, right? I mean, this is something where because the system is so complex, you cannot just write a couple of equations and that will explain everything. Especially things like disease, there is just so much complexity, so much intricacy, so many different ways on how things could develop. So, like Kimberly was saying earlier, you really need a computational model to do that, to do particular applications. And so I believe that by combining all the different types of experimental data and the realistic models, we'll be able to build a platform for computational prediction, right? Where you have a hypothesis, you test it in the model, and then that allows you to provide some direction for future experiments. So I believe this, back and forth between the experiment and modeling will be the most fruitful, and I really look forward to it. And it's happening already in many cases, of course, like including some of the people here. They have done wonderful work there.
I want to put in another plug for rational pharmacotherapeutics, that we can now begin to think about developing models that will help patients. And I think the clearest example of that would be in the severe epilepsies of childhood, where you can identify mutations, you can measure the mutations in a frog oocyte, and then take that into a model and say, well, this kind of drug or this combination or this drug cocktail hitting all these different sites may, to some extent, certainly not entirely, alleviate the severe problems the child's having.
So I might just pipe in that I see that computational modeling actually is incredibly valuable as a guiding post for the science itself, and to also help us understand how should we be measuring. So this gets back to what I mentioned at the beginning, but I think that if we look at it as a data integration strategy, that the data should be informing an integrated picture of the whole, and that we should be using it very much, as Anton was saying, we need a test bed, we need a way of testing hypotheses and theories, but what we need to be able to do is we need to know in an informed way how to measure data that can be integrated. And for me, that's, I would love to see the future that we really have a platform for continual integration of data from neuroscience, and that we know how to measure things in a way that they can be fit together to better inform and test hypotheses and accelerate neuroscience.
I would just say we need to really keep pushing on computational and mathematical literacy of, experimentalists, even just kind of a baseline, so that we can make these collaborations possible. And I think that's really important. But I'm extremely optimistic looking at, the way the field is going. Yeah.
Great. Thank you, Kimberly. Thank you, everyone, for your time and for joining us. And thank you to the audience for all your questions. Have a great day.
Thank you very much.
Thank you.
Thank you.
Bye.